Thanks for being so patient, everybody. I know y'all been waiting for some new information. I've been rolling all over the country, doing events, getting ready for the next retreat. It's been a lot of stuff going on, cooking, everything like that. Also, I had a little problem with my other camera too, so that's why I was just uploading through Instagram, social media, things like that. But I am back. And we're going to get this show rolling. I'm going to have this series. We're going to be dropping these videos throughout the week again so I could bring y'all up on game. Yo, I had to touch on this subject because it's been years. People been coming at me left and right on the viral tip. KT, HPV, KT, herpes, KT, AIDS. What's up with the virus? What's up? Can you help me? So, as always, my best way of helping everybody is by bringing them up to speed. You know what I'm saying? My thing is, you could make better choices for yourself if you understand the mechanisms. When you know how the engine work, when you know how your brakes work, when you know how your oil change and all that goes, there are simple things that you could do for yourself without calling a mechanic. You know what I'm saying? So if you get a better idea about how it works underneath the hood, then you can make those particular changes to help you, family, friends, whatever, So as opposed to just outright saying do this, do that, do the third, it's like you're never gonna learn nothing. So I figure I go into detail on breaking down these viruses, but most specifically touching on herpes, since it's something that can't be cured. The interesting thing about the can't be cured thing is the fact that herpes has medication but no vaccine. HPV has vaccine but no medication, but then HIV got both. A lot of weird stuff going on. People are told that there's no cure for it. My thing is, if you contract anything, if anything within inside you starts to break down, you have the capacity to build that thing back up because you are the healing factor. I am putting the disclaimer out right now. This video is not to treat anything. I'm not saying I'm curing anything. I'm not saying I'm the answer. None of that. This is information and based on scientific study, evidence, experiments, so forth, so on. I'm providing to you the information so you can make those decisions and choices on your own. Okay? But when you see for yourself, you're going to know what to do. So the name of this presentation is Gone Viral. My scientific explanation on HSV, which is herpes simplex virus and other viruses. All right, because when we're talking about herpes, like that's just a name, okay? It's a gimmick. What is it though? It's a virus, okay? And what pulls all viruses together? There's certain information that if you understand about one, you can apply those principles to others, okay? Now, before we go deep into herpes, I gotta talk about this word right here, pleomorphism. Pleomorphism is the word right now, okay? Because in my last few videos and last few interviews, I've been putting forth the information I've been on for some years now, and that is the fact that the reality that we live in regards to healthcare has to deal with germ theory, all right? Germ theory, meaning Louis Pasteur ended up winning the battle against who? Antoine Béchamp, okay? Antoine Béchamp was all about pleomorphism. He was all about water is life. He was all about, it's about the environment that you living in, not so much about, oh man, you have to disinfect, your blood is sterile, everything like that. So because of Rockefeller losing out on all of his contracts because he was an oil tycoon during the time where Edison came forward, he had to find a way to dump all his petroleum, so he figured out, with the whole big pharma thing, to convert all of those chemicals into pharmaceuticals. All right, but in order for that to work, guess what? Louis Pasteur had to win. Vaccinations had to win. You know what I'm saying? So us being born, in the 1900s, 1980s, 1990s, 2000s, so forth, so on, we're not privy, you know what I'm saying, to a world prior to Louis Pasteur. We're in a health world where Louis Pasteur, his word and everything is solid, pasteurization, everything like that. But when you look at Dr. Sebi and you look at the other elders that have brought forth the information, you can see everybody's mentality was on pleomorphism, which means, you know, many forms, transforming into many forms. Meaning, you yourself can induce viruses, you yourself can induce bacteria and fungi, and they all manipulate, you know what I'm saying, from each state to another inside of you. Okay, one of the best ways to explain this is through mitochondria. Mitochondria got its own genome, it got its own DNA. It could replicate on its own, it could fuse, it could do whatever it needs to do, you know what I'm saying. By it having its own genome, what does that mean? That means it's its own organism. Matter of fact, it's like a bacteria, okay? Rickettsia is one of the families that mitochondria would have to be the most closest to, you know what I'm saying? So this endosymbiosis, this relationship, you know, that we have with mitochondria, we providing them the oxygen, they providing us the liberation of the energy, the water, and us being able to work together allows us to have this dynamic existence. The dynamic existence that we currently live in is one in which we are walking solar systems, we are walking galaxies, we are walking universe, okay? Because if you go out and you look at the world, you look at Earth, you're going to see all these ecosystems. You're going to see deserts, you're going to see tundra, you're going to see rainforests, okay? They all have different organisms, different plants living in there. And based on the different plants and different animals living in each one of those ecosystems, that great diversity is which makes up the ecosystem, which makes up that world. It's the same thing with our body when it comes to bacteria and other microbes, especially in our gut. The 100 trillion bacteria that live in our gut, the diversification is what keeps our well-being going on. See, when you decrease diversity, when you take away the lions and you take away the hyenas and you take away the crocodiles and you take away the elephants, you know, the only thing you left is with some zebra and gazelles and a bunch of grasshoppers, you know, that whole environment is going to become imbalanced. You see what I'm saying? Based upon you pulling out key organisms that live inside that ecosystem. So you got to think of the same thing inside your body. So we have all types of organisms working in tandem, working in unison, working in cooperation with one another in order to see fit our reality, our one body. Our one body, if broken down, would just become all types of species. Okay, so we have to understand that. By understand pleomorphism, that gets rid of vaccinations, that gets rid of a lot of the premises that Western healthcare is still on today, that is just so like medieval times, old school, and you throw it out, throw it in the trash. Now, one of the things that blows up pleomorphism is the fact that we all get chickenpox when we're young, okay? Whether we're exposed to it or not, we induce chickenpox within inside ourselves. We are able to express certain particular genetic factors within inside our body that have us express certain viral agents that then gives us symptoms. So when we children, we got chickenpox, and guess what? When we get older, we still have chickenpox in us. They tell us there's no getting rid of it because it ain't about getting cured. How you getting cured of you? You see what I'm saying? That's the trick of the words that they're saying. Of course you can't be cured, because it's not about being cured. It's about something that you are and whether or not you're expressing that side of you. Just like there's a stinking part of you, right? There's just a dirty, trashy part of you that only comes out based on you getting stimulated in certain ways. People doing certain things to you allows that part of you to come forward, or else, as long as everything's cool and everything's Gucci, like, it gets suppressed. You're not that way. You see what I'm saying? So when you get older, chickenpox is shingles. You know what I mean? You got Epstein-Barr virus, you got cytomegalovirus. All of these particular viruses are inside this herpes family, okay? And depending upon your age, your factors, your environment, the stimulus of the environment that you live in determines what gets expressed inside, determines what state this particular virus is in. So when somebody has herpes and they're trying to get rid of it, having a better understanding of what herpes is and how it's affecting you will then give you a better understanding of the steps you need to take in order to rid yourself of such a thing. All right. So like I said, you got to picture the mitochondria, the mama, you know what I'm saying, organelle slash, okay, endosymbiotic organism inside our body, which through its electron transport chain is able to liberate ions and provide us with the energies of protons and electrons. Okay, so we're gonna keep on moving forward. All right, now when you're looking at the pleomorphic cycle, this is just a basic rendition of that cycle, showing how we have a basic unit of life that is not the cell. The cell is actually something more complex. Okay, there's a unit of life that comes before it that we have in a lot of organisms, called somatids. They cannot be destroyed, you know, no matter the pressure, the heat, whatever you apply to them. They live almost indefinitely, you know, based on all experiments. They go through this kind of 3-step cycle. Okay, this little 3-step cycle. Now with this 3-step cycle, as long as it keep going in that 3-step cycle, everything's all good. But when it progresses into the 4th, the 5th, all the way through the 14th, that's when we have bacteria, that's when we have viruses, spores, fungi, you know, yeast, mold, all those types of organisms ends up developing. And one of the main factors that promotes the upward development of these microbes is the pH of the body. So as pH decreases, meaning the increase of acidity, then what we have is a forward movement of the development of all of these microbes. Okay, now of course you have your microbiome that consists of bacteria. You have candida that's inside the mucus lining, you know, of your alimentary canal. So you have circumstances where a lot of these particular microbes are in certain stages, but they're still in harmonious, non-pathological stages. The pathology comes through the explosions, the overgrowth, things of that nature. When somebody wants, you know, 3rds, 4th, 5ths, you know, with dinner as opposed to just seconds, they trying to get a bigger piece of the pie, you know. Now, when you're dealing with herpes specifically, you're dealing with different stages. All right, so we have attachment, which is when the virus binds to the host cell. Your cellular structure is the host cell. Penetration, okay, virus injects its genetic material into the host cell. Virus is like males, bacteria, host cells are like females. So you have viruses that don't have the machinery to replicate on its own. So as far as the bacteria, a host cell to what? Penetrate and inject its genetic contents to do what? Fertilization or the unification of DNA, so that the mechanisms that make its body up, the protein that makes up its caspid, its envelope, things of that nature can be produced so that it can be produced and synthesized in vast numbers and take over the body. Okay, you got uncoding, meaning the viral contents are released. Biosynthesis, the viral genetic material replicates via the host. Assembly, viral components and enzymes are produced and begin to assemble. Release, newly produced virions are released from the host cell and move on to infect other cells. Now, these steps are broken up into pretty much 2 cycles, which is called the lytic cycle, okay, and the lysogenic cycle. Okay, the lysogenic cycle is the process in which the host cell is replicating, and in the host cell replicating, it in proxy is replicating the recombinant DNA created between the host cell genome, your DNA, and the virus DNA. Now that is latency. That's when the virus is asleep. It's not no outbreaks, anything like that. It's quiet. It's just your cells are replicating what the blueprints, the instructions. Now, the lytic cycle is the process when replication starts up and the actual recombinant DNA starts getting transcribed, and that transcription is translated into proteins that then are assembled in order to make the individual components that make up the virus. So the lysogenic is kind of the latent, kind of hibernating side, but then when you got the lytic or the lysis, the lyses, like hydrolysis, you know, that's actually the active component of it all. All right, so in order for us to understand Viruses, we got to understand DNA, because that's all viruses are. It's just foreign DNA encapsulated in some type of envelope, all right, in some type of protein covering, all right. And to start with DNA, we're going to start with chromosomes. Everybody know what chromosomes are. You got 23 from your mama, you got 23 from your daddy. Put it together, you get 46, you got a human being, right? Now, out of them 46 chromosomes, there's sections of that chromosomes where you will find DNA, and in that DNA, you'll find sections of the DNA which you will find genes, and based on the transcription or translation on that gene will determine what protein, what enzyme, what thing your body is going to create. Okay, but before we get into all that, we're going to start with chromosomes, and chromosomes are like zip files. You know what I'm saying? Like, I just filmed about 8 hours of footage. You know what I'm saying? My man's Keith all the way up in Detroit. I need my footage. I want to do editing. He can't send all them gigabytes down to me, so you got to zip that bad boy up. You got to compress the file. Send the compressed file to me. All right? Now, when he sends the compressed file to me, when I receive that compressed file, I unzip it, and in unzipping it, it unleashes all of the particular footage. You know what I'm saying? Now I can work on everything I need to work. So DNA, you have an idea of a double helix. When you compress that double helix, you end up getting these particular figures, these X and Y shaped figures known as chromosomes, which are nothing but compressed versions of DNA. Okay? Now, DNA, deoxyribonucleic acid, okay? This is the double helix right here. I'm going to get into a couple analogies because I know you hear this term, but you don't really get what it all is, okay? You got two strands, and you have steps to your ladder. Same way you got a ladder, just imagine that you twist it around into a helix. You got one side of the ladder, the other side of the ladder, and in between the two strands are steps. These steps are bases: cytosine, guanine, adenine, and thymine. All right? Now, think of DNA like a hard drive in the computer. Okay, it's a source information. If I need anything from the computer, I'm pulling it out of the hard drive. That is what the DNA is. I'm pulling it from the DNA. Okay? Now, a section of that DNA, if you notice this DNA right here, this DNA is nice and long. Okay? Nice and long. When I pull a section of that DNA out, though, that's a gene. And a gene is just a section of the DNA. That section is equivalent to a file, all right? And that file is a file for a blueprint, all right? So gene will equal blueprint file, all right? Now, in pulling that file, okay, I want to copy it, because anybody who's dealing with a computer and dealing with a hard drive, and you need to do any work, you're not going to pick the computer up and take it with you. No, you need to copy that file. So the mechanism that you use to copy the file to take somewhere else, all right, is known as RNA polymerase, which is an enzyme, okay? But the mRNA, meaning the actual copy, the file that you're pulling from, the copy of the file, all right, can also be equated, like I said, to a file or email. And the USB is the mechanism that you can use to carry it somewhere else, okay? So think of it as a USB that's holding a file, holding a copy of a file. Now you can take that file to any other computer, to any other mechanism, upload it, print things out, see what the display is, so forth and so on. So a gene is a section of a DNA similar to a file, email. mRNA, messenger RNA, is a copy of that section, and it allows the transport of the copied information. Okay, so we got source. I need to make a copy. I make a copy. I'm taking a copy somewhere else. All right, this is what's going on when you're dealing with transcription, translation, and protein synthesis. All right, the dogma. Now, this is RNA polymerase. It's a word, don't be scared. RNA stands for ribonucleic acid as opposed to deoxyribonucleic acid. The difference is it's a single strand as opposed to a double strand. All right, polymer. Polymer just means links of molecules, okay? In this case, nucleic acids, all right? And ACE is a suffix that denotes we're talking about an enzyme. Enzymes, what speeds up chemical reactions, which would normally take, based on nature, 100 years, could be shortened down to 5-10 minutes, okay, or less than that. All right, so if you notice in this picture right here, the brown object is the RNA polymerase, okay? The blue and purple represents the DNA. If you notice, it's pulling the DNA apart or unzipping the DNA, and it's creating another strand, mRNA, the copy of the file that is copying from the source to use in order to synthesize a protein. All right. So what RNA polymerase does is it unzips the two strands. I know you've wore a jacket before, or some jeans or some pants, and you've had to use a zipper, a purse, something, right? You have two complementary sides. They both teeth, but the teeth are not the same. They interlock, and they interlock based on that zipper. That zipper come up is zipped. That zipper come down is unzipped. All right. So what RNA polymerase does is it unzips the strand. So RNA removes the hydrogen bonds. This action splits the complementary pairs apart from one another, creating two single strands. One strand is used as a template to form a new complementary single strand, which is the file, the mRNA, before it is reunited with the original strand it was separated from in the first place to reform the original double strand so it could go back to what it was. So it's like you got a file, you got the double strand, you unzip it, you make the copy, and you put it back how it was, you know, before. And if you notice here, you see the splitting. All right, and it's just split by removing hydrogen bonds, because hydrogen bonds is what keeps the complementary bases together, the same way that the complementary geometry of the teeth on the zipper keep them linked together. All right, so if you notice on this picture, you see that dark purple is the growing strand. The light blue is the double strand of DNA. The light purple is the polymerase as it's moving down, unzipping the DNA, creating a complementary strand off the split strand that it has inside. You see that purple. The little orange specks that are floating around in the picture, those are floating nucleotide bases, either cytosine, adenine, guanine, or thymine, that the RNA polymerase is utilizing in order to do what? To build the mRNA, to build this file. All right, so when you deal with drugs that's for herpes, like what are they doing? All right, so they act as decoys that are inserted in the viral DNA during synthesis and stop the elongation of the viral nucleic acid, thus temporarily suppressing the viral infection. Since these drugs target viral DNA synthesis and the ever-changing viral enzymes, they are prone to drug resistance in the long run. All right, so what they're saying is, in this process right here, where the RNA is actually, the messenger RNA is actually getting created, what happens is they actually create decoys. So those little orange nucleotide bases that you see floating around, that the purple, the light lilac colored RNA polymerase is, you know, accumulating in order to grow the file or grow the mRNA. The drugs are actually decoys or fake synthetic components of those orange nucleotides. So as it pulls those in, it gives a false message that the mRNA is done, that it's finished, that it's finalized. So it finishes it, and because it's an incomplete message, it can't go on to being actually translated into a protein that your body's going to end up using, or the actual virus is going to end up using. Okay, here's another picture of the RNA polymerase in the light green, dark green. The rainbow color is the DNA. You see that they're actually making an RNA strand in the bottom right of the screen, right above the sign. Well, the double helix is on the top of it right there, and then you see double helix in the background. All right, so the gene is instructions to build. So liken a gene to blueprints. Okay, a gene is blueprints. mRNA is a copied file. Okay, use a USB in order to store the copied file, but the file is of a blueprint, and that file is a gene. It's of a gene. Okay, now in construction, the contractor interprets the blueprints. All right, then what? Instructs the head foreman, all right, who's over all the construction workers, the head guy, And then the construction workers in your body are known as ribosomes. And the ribosomes assemble and build. So like how you see him doing the roof and the steamrolling, stacking up the bricks, mixing the cement, whatever it is. Your ribosomes take that mRNA and they end up now synthesizing amino acids into a chain link to develop into a protein. You know, and a protein can be anything that your body uses as a tool, a hammer, a saw, a screwdriver, anything of the sort. All right. So here's a picture of the ribosome. The long string is obviously a mRNA. Okay. You got two components that make up one ribosome. You got a large subunit, a small subunit. They come together, they coalesce on the mRNA. All right. Here's another picture right here where you have the arrow is pointing to tRNAs. This is another form of RNA I'm about to speak on very shortly, called transfer RNA, which actually holds an amino acid on there. This is the transporter that brings the amino acid over to the ribosomes. All right. So here's a more simplified picture with the ribosome on the mRNA. mRNA are the colorful little lines that are coming from the strand. If you notice, if you look real close, they're in threes, okay? And that's called a triplet codon. Triplet codon. Triplet codon could be maybe AAU, which means adenine, adenine, uracil, or it might be GCA, and that's guanine, cytosine, adenine, you know. And what is that for? Well, because they have these triplet codons of these individual base pairs, when that transfer RNA comes over with the amino acid, it has a complementary triplet codon that it will fit to. So it will only fit to the three that it's going to complement. And then that unification of the two complementary pairs allows a release of amino acid by way of the ribosome we're going to see shortly. So here's a better picture of a transfer RNA, tRNA. And right here we have the amino acid on the upper left, and then on the bottom we would have the triplet codon. All right. Take a break. What are we on? We counting down. 3 more minutes. Okay. 3 more minutes. Amino acid is on the upper left corner. Triplet codon is on the bottom. So like if you see on this right here, it's going to drop that triplet codon on there. But let's move to the next simplified picture. Once again, on the bottom you got the 3 bases that are going to be complementary to the 3 bases that's on that mRNA. All right. Then you have the amino acid on the top. All right. So what ends up happening is each one of these tRNA meet up with a complementary base pair, and as they meet up with the complementary base pair, they release an amino acid, and they do it in succession until a string of pearls start to get made, because they keep on coming. All right. And that's what this is. Now you see E site, P site, and A site because those are all sites in which something's happening. As the tRNA lands, it lands in one site, and then it shifts to another, and then another one drops in, and then they shift over, and then another one drops in, and it shifts over. It's like a factory, and it keeps going. All right. So in this process, as this ribosome, if you notice, it doesn't move down the course of this mRNA, you know, like a track. It's now producing a polypeptide chain, which is the prerequisite to a protein. It just has to get folded into the necessary shape of whatever tool is going to end up being. But if you notice, it's slid down, it's going through this process where tRNA is meeting up with them complementary base pairs, stringing them amino acids up. Now you got a polypeptide chain, which is now going to end up becoming a protein. Okay? The finished home, because remember we were talking about ribosomes being construction workers, when they're done building the house is the protein. Okay? And that is the process that we go through, you know, in order for transcriptions, translation, and protein synthesis to occur. And the reason why we're touching on this is because in order for you to have an infection or an outbreak, This has to occur because viruses need new parts. They have to multiply. They're not multiplying without this particular process going on. So I wanted to make sure that y'all had this in a simplified form so y'all can get that understanding before we dive in deeper waters shortly. All right, so as I mentioned before, the finished product is the protein. So the finished home is the protein. All right. Now, if there's misspelling, all right, if there's misspelling that occurs, that would equate to a mutation. And in that means the final product is not going to be sound. It ain't going to be a sound final product. As you see with these puzzle pieces and just the integrity of this home, it ain't nothing like this home right here. Okay. So a mutation on a transcription translation of a viral body is a good thing, because that means that the quality of the envelope, the quality of the capsid, the quality of one of the parts of the virus is going to be subpar, is not going to be good. And based on that, that means it's easier to take it out the game. So that's a good thing when it comes to that. All right, so let's get into the herpes. So herpes falls into the icosahedral virus families, okay? Because you got a lot of viruses and they have different forms, and the herpes virus is very interesting because not only does it have a capsid, which is this envelope, this icosahedral envelope. Icosahedron is one of the platonic solids, the chemetic solids. Let me get that right. And we have different chemetic solids. We got the tetrahedron, the hexahedron, the octahedron, the dodecahedron, and then of course you got the icosahedron, icosa dealing with the 20 faces. Now this particular icosahedron is implemented within the sacred geometry of these viral bodies, these protein wraps. These are the membranes, the skins that do what? That hold the genome, hold the DNA, hold the instruction, hold the file. Okay? These are the USBs. These are the sacred geometric USBs that hold files. So these USBs can just go wherever they want to, plug into a port, and do what? Upload, download their contents into another server. All right, so we got the right foot motif, over there. You got the single barrel motif. Okay, that deals with what? Papilloma. So that's going to be HPV. Okay, then you have these over here, the double barrel, and these double barrels deals with the adenovirus, common cold. And then you have the swan-like motif that's actually dealing with the herpes family. All right, which is in this upper left corner. I've been saying right, but this is what, upper left. All right. So this is another virus right here, the chikungunya virus. This is spread by way of mosquitoes. All right. Look at its geometry. Look at its architecture. Look, I picked these pictures specifically because I could come up here and say a whole lot of big words, sound all fancy, whatever. But the idea is for you to understand what you're dealing with. Okay. The geometry on the microscopic levels, the nano levels, the quantum levels, it's just amazing. Okay, like when you look at the way these things are put together, you find why there's so much intelligence involved, because this ain't no fly-by-night structure. This is real sound ingenuity here. Okay, and you're gonna notice that the yellow represents the capsid surface, okay, which is made out of glycoproteins. And inside, underneath the pentamers means little facets of pentagons, little five-sided facets that make the capsid up. Underneath that's all there to protect the genome, the genetics, the file inside. I mean, you got a USB to do what? To protect the file. You got a memory card from your camera to do what? Protect the file. You have a terabyte to do what? Protect the file. Because if the file was just out on the black carbon or whatever like that, it can get affected, it can get erased, it can get destroyed, it can get messed up, so that when you try to pull the file out and print it now, it's only half the paper, or the color's missing. You know what I'm saying? Or it's something totally different, or it's not there no more. Equate that to genetics, okay? All right, so the human papillomavirus, HPV, once again, here's the structure. It's split in half. All right, it says the virus infects human skin and mucosal cells and causes warts. You know, it's real bad with the cervix. You know, it's a main cause of cervical cancer. You know, of course, sexually transmitted. You know, and everything like that. But understand, based on pleomorphism, you have the ability to induce a lot of these things on your own, based on what? Light pollution, based on what? Food intoxication. Okay, the way you're living your life, your standards, your hygiene, and where your environment is. You might be living in a city like New York where you're right in Manhattan. I'm talking about lights all day, Times Square, noises, sirens, smoke, all types of stuff going on. You mean to tell me that is not going to affect your genome? You're going to induce all types of stuff. You know what I'm saying? So yeah, maybe the girl didn't have this, or maybe the guy didn't have that. But just because of the environment that you in, the way your mind is at, where her mind was at, what y'all eating, what y'all on, y'all can induce the expression, okay, and ignite the expression on volatile bodies inside of you. Because when I showed the pleomorphism, the whole thing about the somatids, the main thing about those little units of life is they know that your spacesuit is on loan. And when I say spacesuit, I'm talking about our human body. The body that we use is on loan, and it has to go back to the earth. You see what I'm saying? When you're done with it, based on the decisions you make and how you live your life, your body gets signals and gets word whether or not you still want to live. And when their signals is telling it that you want to die, you pro-death, you ready to go, it's going to start breaking the body down. It's going to do by whatever means. And if it means it has to upward regulate, you know, molds and bacterias and yeast and viruses in order to take you out the game, they will. Okay, you will. Correction, you will. All right, here's the common cold, adeno, right here. All right, anytime people got their little cold and they feeling what they feeling, this is the guy. You know, you got HIV, you know, human immune virus right here. You see the complexity of its external envelope. You know, the cone, it has like a cone shape caspid inside. It's a cone-shaped caspid deep inside, holding the genome, and you see its glycoproteins, you know, on the surface are very, very specified to do what? To evade your immune system. All right, here's influenza for all the flus. Okay, this is the influenza. You know, you start sneezing and the sinuses get all messed up, and you know your temperature shoots straight up. This is the particular organism right here, or not organism, because they say they ain't alive, right? And they ain't dead either. So what are they? The green on the surface of the glycoproteins, okay, you have yourself a capsid, okay? It doesn't have an envelope and capsid, similar to the papillomavirus is just the capsid, the adeno just the capsid. There's only specific viruses that have, you know, another membrane around it, you know, such as herpes. All right, this is just showing the particular pentamer as well as hexamer mosaic that covers the surface of the HIV. All right, here's the HK97 fold, okay, which is a geometrical term of this particular capsid, which lies inside of the envelope of the herpes virus. Okay, so herpes has 3 stages, okay? Within 18 hours after infecting the cells, HSV makes 3 types of genes: the immediate, early, or alpha genes, then the immediate or B, beta, and then finally the late or gamma genes. You got alpha, beta, and gamma. For a successful infection, these genes must be made in that sequential order. They do not go gamma, then beta, then alpha. They do not go beta, then alpha, then gamma. It's A, B, G, okay? Now, for a successful infection, they have to go in sequential order. The alpha gene, ICP4, okay? ICP stands for infected cell protein. plays a pivotal role in its viral cascade and is an attractive therapeutic target, since without its expression, all early genes cannot be transcribed. So my thing is, when you're dealing with herpes and overcoming it, then a lot of people, even with the medications, they deal with coming in on the beta and the gamma stages, as opposed to dealing with the alpha stage. My thing is, deal with the alpha stage, hit it in the alpha, because if you prevent ICP4 from expressing itself, then guess what? None of the other ICP47s, 27, 22, all them different infected cellular proteins that are going to start doing its role, you know, in regards to promoting the progress of this particular herpes virus inside of you, all of it's going to get shut down. All right. Now, here is the structure of the herpes virus. Okay, so that capsid that you saw before right here sits inside an envelope, which is white. Okay, and that envelope has those little white little studs that are sticking out from it, and those particular studs are the glycoproteins. These glycoproteins are what are responsible for what? Getting the virus inside of the cell. Because remember, before you talk about replication and warts and issues and blisters and lesions and all those things, that's based on replication. That's later. Okay, he got to get in you first. Okay, so that's why it's like, okay, stick with the one. That's what all the Moors is definitely on in regards to, you know, how they deal with law. They like, yo, when you deal with these court systems, when you deal with all of this stuff, stick to the one, the original, the jurisdiction, all that. You want to deal with it getting in you in the first place. Just like a woman, it's like they don't have to think about none of that other stuff with sex. It's about, okay, did he penetrate? Did he even get in there? If he never even got in there to begin with, then yeah, what are we even talking about, right? So the envelope of the herpes vaccine is covered in arginine clusters. Arginine is an amino acid that plays a huge role in herpes expression, replication. Okay, now you see the pentamer shape right there, and the little six-sided flowers that are in there. That's orange, beige, and blue. Those are just showing the different protein structures that are actually formed into geometrical compounds that make up the pentamer, or this pentagon 5 shape, and then the pentagons fit together and they end up making, okay, the icosahedron caspid shape, you know, that holds in the virus. But the only way that the DNA, the genome of this virus is going to get in you is if it gets in you. Okay, so let's get on that. Now, as I move through this, I'm going to give information on natural mechanisms that exist around us, all right, that are there in order to assist us when we have an upward of a viral load, explosion of viral loads. Okay, for instance, you work out, you know you're gonna smell a little something, you go wash up, you smell better, you put on deodorant. You know, if you don't put on deodorant, you're gonna smell a certain way. Might not be that bad, but throughout the day, if you keep going, it's gonna get progressively worse. And if you don't wash up for the next 2, it's gonna be funky. You see what I'm saying? So you gotta wash up. So we always got viruses, we always got yeast, we always got bacteria. Like I said, we are walking universes. It's all about keeping the balance, okay? Because if the Martians is like, yo, this is what's up, we about to take over the solar system, and the Venusians is like, nah, I'm not playing that, we about to like take this thing over, you know, then the Jupiterians and Uranians of the solar system, they're gonna be sitting back like, what's popping? There's gonna be a war that's gonna go on, and everybody suffers, okay? And that's what goes on inside of us. Now, when you look at a plant called carambola, all right, carambola, also known as starfruit, it has a ratio to 2 particular amino acids, and these 2 amino acids that I'm talking about is lysine and arginine. All right, when someone has a viral load inside of them, specifically herpes, Arginine plays a huge role in the replication and the survival of that virus in your body when arginine numbers are through the roof and when lysine numbers are low. Okay? So arginine bioavailability is absolutely necessary for the replication of herpes simplex virus, which causes cold sores and genital herpes. When arginine is not available, herpes viruses and cells are unable to complete a single replication cycle, and cell damage is evident in infected cells. Arginine is not involved in the early steps of viral replication at the level of viral DNA synthesis. Rather, the amino acids is necessary for the expression of the late viral functions, such as the synthesis of the viral coat, you see, because like I said, they make up the clusters on the envelope, and production of complete infectious virions. All right, so that comes later. So you want a certain ratio in your body. You do not want your arginine levels to be higher than your lysine levels. This fruit, this star fruit right here, its lysine is 497 milligrams, okay, and the arginine is 135. So the ratio is 497 to 135. That's a crazy ratio. Now you'll find through some scientific studies now that lysine is not everything. Okay, and let me explain. All right, so based on this, you know what I mean, you would think, okay, I just got to take a bunch of lysine and I'm good. No, not exactly. Not exactly. Okay, so ratio is key. Lysine limits the in vitro replication of many viruses, including HSV. Okay, when you talk about in vitro and in vitro is outside the body, cultured medium, in vivo is in the body. The concurrent depletion of arginine is essential for lysine supplementation to be effective. This finding suggests that lysine exerts its antiviral effect by antagonism of arginine. Arginine is an essential amino acid for viral replication, but in the presence of small amounts of arginine, lysine supplementation reduces viral replication by about 50%. However, this effect was not seen in the media containing high in arginine concentration, suggesting that the high lysine arginine ratio is critical for efficiency. Now, this is a picture of lysine. That's a picture of arginine, amino acids. So what's pretty much being said is, it's not just about not having no arginine and eating a ton of lysine. No. It's about the ratio of arginine to lysine. The lysine is going to be effective in the inhibition of viral replication contingent upon you having low levels of arginine and then having a higher level of lysine with that. If you just strictly go off of taking large quantities of lysine and you got large quantities of arginine, what's going to end up happening is you're going to promote replication. Lysine is actually going to end up becoming a promoter of the replication. Yes, the lysine will be like the arginine in that case. You see what I'm saying? So if it sounds confusing, what I'm saying is don't go out your way just taking a bunch of lysine, because the important part is to bring your arginine levels down first. That's most important. You don't want high arginine and then dump a bunch of high light. No, you want to bring arginine down. And in you bringing arginine down, then you want to make sure when you're consuming that the ratio is in favor of the lysine rather than the arginine, and in that you'll end up inhibiting the replication. Now this is just lysine arginine ratio. There's other mechanisms at hand here. So just so you know, lysine deals with the prevention and the assistance of overcoming things such as Alzheimer's, which you're going to find by the end of this how that ties into everything, and dementia, hair loss, shingles. Why shingles? Shingles, herpes, same thing, right? Cancer ends up getting produced based upon another herpes, the Epstein-Barr as well as the Megallo, you know, and then cardiovascular diseases and aging. All right. Now, In order to understand herpes, we need to start with this glycoprotein surface. The skin of the particular herpes has these glycoproteins, and these glycoproteins are what are responsible for getting the actual virus into the cell. All right, so here's a blow up right here. All right, this is a blow up of the influenza. Okay, virus. Viral entry or viral fusion is facilitated by the surface glycoproteins of the virus capsid membrane and the heparin sulfate polymers that extend from the core proteins in the host cell. So, viral entry or viral fusion, okay, because what happens is the two membranes fuse together and the virus gets let in. But the only way that happens is based upon the facilitation is based on the surface glycoproteins, which are the green nodules that are sticking out of the gray membrane of this influenza right here, where the green arrow's pointing to, all right, on the virus, and the heparin sulfate, which are these other mechanisms that stick out of the surface of the host cell, meaning you or a bacteria that is trying to invade. All right, to give you a better understanding, this is an image, okay, of a virus. The colorful geometrical shapes represent the capsid inside. The white envelope around it is the mechanism that's going to allow this virus to enter inside of the host cell. The host cell is represented by the white bottom. Okay, so if you notice, that white bottom has things sticking up as well as the envelope has things sticking out. Here's a blow up of the Lassa virus to give y'all another angle. If you notice here, this represents the surface of the envelope. You notice those colorful mechanisms extending down from the envelope, and then you have mechanisms coming up from the surface of the cell. These things have to link together. They have to lock. A signal has to take place. Communication has to take place. Like if I was going out of town and I had me a cart, what do they call a trailer, right? I have to put the trailer on the hitch. If I don't put the trailer on that hitch and get that thing on right, then when I drive off, that trailer is not about to trail me. Thing about to sit where it's at. So you gotta hitch these things. Gotta hitch what's on the surface of this virus onto the particular surface of the cell. I'm going to show you what I mean. So this is a virus at the top of the screen in red with its surface proteins in yellow, and on the bottom the blue sticking up represents the surface of the whole cell. And if you notice, as it gets close, they lock into place. Once they lock into place, they send the information to do what? It starts communicating to the phospholipid bilayer of the cell membrane, which is represented by the little blue things with the little legs. They're facing up and down. Those are the lipid bilayers separating waters from waters. The green represents the anchor, right? And if you notice, it bends at an angle and it pulls, it splits the bilayer into single layers. It incorporates the single layer into its own layer, all right? And then it fuses with that layer. Now that yellow cone shape is the capsid. The capsid is where the DNA, the genome of the virus is. As you see, it's now entering into the cell because the viral membrane became part of your membrane, and now you can't even tell the difference between the two because now it's inside of you. All right, so that is how the viruses are able to enter. Okay, so you have to deal with what? Manipulating this part right here, that attachment. Because if you can stop that from happening, then none of these sequential parts are going to end up following. None of that can happen. Okay? So when we come back shortly, I am going to continue by talking about how you avoid all these things with the natural components that exist around us. All right, so before I showed that the main mechanism that takes place in the onset, in the early stage, is this virus entering inside of you, okay? And they enter inside of you because on the surface of the envelope of the virus, has particular glycoproteins that attach to surface proteins that are on the host cell. And if you see in these slides right here, as the virus approaches the surface, they lock together. In the locking together, they manipulate your membrane to open up and fuse with its membrane, and in it becoming fused with its membrane, it can now enter inside your cell. All right, once inside your cell, it gets wrapped up by particular proteins, and it manipulates these two motor proteins that exist inside, called dynein and kinesin, okay, or kinesin. Dynein is minus, it moves towards the minus end of microtubules, while kinesin moves towards the plus end of microtubules. In other words, dynein is a one-way, kinesin is a one-way street. The one-way street is from the membrane of the cell to the nucleus. Kinesin plus meaning one-way street going from the nucleus to the membrane. So if you notice this structure, it has legs, and the legs walk on the microtubules and carry the virus where? To the center of the cell, to the nucleus. Okay, the nucleus. And this is the nucleus right here. Blow it up. The nucleus has what? What feature do you notice about this nucleus? It has holes, and these holes are called pores. That is the pore of it. The pores are there because they are there to be selectively permeable, to only allow certain things in. Everything cannot get through this pore. It's like a portal. Okay, if you don't have the right charge, the right energy, you're going to get dropped. Now, because this dynein done transported it, and because of the mechanisms of the herpes virus, it's able to manipulate a lot of our signaling mechanisms, and in so trick us into allowing it inside. All right. Now, once it's inside, what's it going to do? It's going to do exactly what I started all this thing off with the whole thing about the gene, the USB, the copy, the blueprint. Now it's doing all of that inside of you. So it could do what? It can leave out the nucleus, and then it can bud. Okay, viral budding is when they end up getting on the kinesin, which goes back towards the surface. You know what I mean? The cellular membrane, and they start to bud from the cellular surface out into the extracellular fluid and the rest of the body, so they can do what? They can repeat the process over and over again. All right. Now you got people who have herpes outbreaks once a month, once every 3 months, once every 2 years, might be 5 years, nothing. All of a sudden they got outbreaks. Like, I thought it was gone. Well, herpes simplex virus hides in the nervous system. This is why shingles, you get the tingle and stingy pain, because it's manipulating and destroying and overstimulating your nerves and the nerve endings. Now, HSV specifically hides in the sacral ganglion, okay, or the trigeminal ganglion. A ganglion is the collection of nerve somas, okay, nerve somas. Let me give you an example of a soma. Do I even have a whole? I don't even have a whole picture. Okay, we'll use this guy. All right, so on this picture of a nerve, the long arm is the axon. That main part, thick part, is the soma, and then all the extensions coming out of that, going from there, is the dendrite. The dendrite receives the information, soma processes the information, axon transmits information, synaptic knob, it wirelessly transfers it. It hops from the presynaptic knob in the synaptic cleft to the postsynaptic terminal to the corresponding dendrite on a corresponding nerve. Okay, now in the soma, okay, when you have a collection of somas of nerves, because your nerves go long in the body, they can go the whole length of your arm, whole length of your leg, but there's only like one soma, one area where the nucleus is at. And when you have all those nucleuses together in one area, that is a ganglia. Okay, now sacral ganglia represents the sacral region, the lower part of your back, while the trigeminal ganglia represents the throat, okay, the jaw, all right, your face. So that's why you got herpes simplex virus 1, you got herpes simplex virus 2. Okay, we all know HSV1 is also known as cold sores. Where do you experience them? On your mouth. Why? Because they hide in the trigeminal ganglion. Then you got genital herpes, okay, where you have problems in your reproductive region, and the reason why that occurs is because they hide in the sacral ganglion behind your genitals. Okay, so they hide there, and they hide there under the pretenses of two processes. One of them is called neuronal restrictive silencing factor. The other one is called human repressor element silencing transcription factor. These are the two mechanisms that deal with latency, when herpes just be gone for you think indefinitely until you do something and it gets to popping. And that's because what? Lysine levels, arginine levels is real low, you know what I mean? And lysine levels is high. You know what I'm saying? You might be just on it. You're doing what you need to do. And it's like, yo, I can't afford to go out there. Like, if the block is hot and you specifically hot, then you know to play it. Yo, I'm going to just, you know what? I'm about to go to the Carolinas for a couple weeks and just kind of lay low till things blow over. You know what I'm saying? And then once things ain't hot no more, I'll come into town and pick up where I left off. Okay? That's what's going on. All right, but that's a good time to attack. That's a good time to really get at them, but you got to know how to get at them. Okay, and if you notice this picture of these nerves, looks like a forest, looks like a tree. It's that dense, you know, and they hide amongst them. You know, they hide amongst them. You know, neurotransmitter. This is synaptic transmission, as I was speaking before. You know, at the end of the axon is a synaptic knob, and the synaptic knob is where neurotransmitters are fired. And if they're hiding amongst the nerves, you know what I'm saying? If they're hiding amongst the nerves, then they definitely can stay abreast on your communication. What you thinking, what you eating, what you not gonna do, what you are gonna do. Getting up in the middle of the night to eat something you ain't supposed to eat, all types of stuff like that. It can tie into that because it's in your nervous system and it's hiding specifically amongst the microtubules. Now, microtubules I do whole presentations on. They're one of the most dynamic parts of the body next to the mitochondria. I really put them kind of hand in hand. They work together. What they are actually fiber optic cables that are made up of individual components called tubulin. These tubulin, alpha and beta tubulin, actually play a role in quantum computation, specifically on and off in a superposition of both. And based on the actual triggering of on, off, or the entanglement of both of them, gives rise to a coding system, a fiber optic signaling mechanism that occurs throughout your body by way of the microtubules. And by them only having a 15 nanometer lumen meaning the space, the cylinder, gives rise to monomolecular signaling of water molecules. What does that mean? Monomolecular. Mono means one, molecular is molecule. So monomolecular signaling, meaning water is able to actually structure itself within inside the lumen of these microtubules, and because of it being able to structure in such a tight space on nanoscale, it has these more refined properties, okay, of for energy and for signaling that occur. Now, if the herpes viruses are hiding amongst these microtubules, then guess what? It can tap in and it can find out what you on, where the body's sending all the zinc at. You know what I'm saying? Where the body's sending all of this at. You know what I mean? Where's this? Where's that? I got to get up on that, right? So the nerves are composed of the microtubules, which I say the real nervous system. HSV hides amongst the microtubules of nerves, especially in the presence of heavy metals during latency periods. Due to their fiber optic ability, it can stay abreast to your communication. All right? Now, these are the mechanisms that I spoke before, the dynein and the kinesin. Okay, they're like legs, all right? And ATP plays a huge role in their walking. On the bottom in the gray is the microtubule, as you would see in this same picture. Okay, that long tube, okay, is the microtubule. The dynein is carrying that big bubble. That big bubble is a vesicle carrying contents. Just imagine that's a herpes capsid holding DNA, and it's walking along, all right? ATP mediates the transfer of energy of easy water, which is exclusion zone water. To give you all a brief understanding of easy water, whenever infrared energy, electromagnetic energy of any kind hits water, specifically infrared though, the water near the surface, meaning the water sitting right next to the glass in a cup, or the water sitting right next to the side of the tub, that water starts to structure itself in a hexagonal lattice similar to what it does when it's ice. Okay, and it's predominantly electrons. It creates a charge of predominantly negatively charged electrons and pushes the protons to the center. So you have this electrochemical gradient. You have a battery. So all water is a battery, with sun electromagnetic radiation being the source. So ATP mediates the transfer of energy of the exclusion zone water by way of quantum entangled phosphate groups from ATP to fuel the power strokes, the walking of the diene and the kinesin, one leg after the other, walking tubulin like the lit sidewalk on Billie Jean. So anybody who's seen the Billie Jean video, and you see how he getting it, and you see how the different squares in the the different squares in the sidewalk is lighting up, and then he hits it, and that's how he doing his moves. It's the same thing with these microtubules here. You know, each one of the tubulin that make the microtubules up, the alpha beta tubulin, they'll light up on, off, or the entangled ones, and that is what lets these particular Dinean know where to put their feet at. Okay, they almost kind of walk like the drunken master. You know, anybody who's tore up slizzard from the club about to hail or Uber, he all over the place. You know what I'm saying? But drunken masters be like that, but they still cohesive. They still know where they're going, and that's kind of how they walk. They walk a little drunk. All right, so once again, this is a cross section of the microtubule, side view, and then the components that make them up, these little peanut shaped components, which are called tubulin, and it's alpha beta tubulin. These tubulins are made up predominantly of aromatic amino acids. Aromatic amino acids are photon traps, waveguides. They absorb photon light, they channel it. So this whole mechanism is a photon signaling quantum fiber optic system. It's amazing. It's amazing. All right, so let's talk about secondary plant metabolites. When we talk about primary plant metabolites deal with growth. They deal with reproduction, survival, the basics. You know what I'm saying? When you deal with secondary plant metabolites, that deals with the things that they use for defense mechanisms, extra stuff, okay? Plant secondary metabolites are similar to the metabolites that we have or we need in our body. The good thing is that they're in forms that we can assimilate. We put it inside our body. Cytochrome P450 systems are able to reduce, oxidize, whatever these things, and turn it into these functional parts that are able to induce or inhibit certain functionalities that we need to be in charge of. In this case, we're talking about what? Viral infection, right? So lignans, okay? Plant secondary metabolites contain a long list of active compounds that influence metabolism, such as phenols, called so because they contain a phenol ring, as in like phenylalanine, which is an amino acid, make a large group including coumarins, stilbenes, anthocyanins, and phenylpropanoids. Okay, now phenylpropanoids can form complex dimeric structures. Dimeric means two. Okay, so when two structures come together, when they use their side chain, so you got an aromatic ring with a side chain, an aromatic ring with a side chain, and when they come together to make a dimer, a dimeric molecule, okay, you call those lignans, and they're the main component of wood. So when you see trees out here growing up 30, 40 feet, branched out 100 feet out this way, 200 feet out that way, with leaves and fruit, and the wind can't do nothing to it, that structure that it got, that integrity is there because of the lignin, okay, the lignin molecules that make up its wood, which they're most concentrated in the areas that hold the most stress, so a lot of the joints. The densest wood in the world is of the Lignum vitae tree, which grows in St. Croix, and it contains the lignin guaretic acid. Guaretic acid. Now, another form of guaretic acid you'll find in a very powerful herb, all right, by the name Laurea tridentata. Big shout out to my queen chakra oracle. Her name is Lurie. All right, so I found it quite amazing that she kept telling me, yo, you got to do that. You got to do the herpes joint. You got to do the herpes joint. And one of the main herbs, you know, that I would have to talk about, you know, they share their titles. I find it quite interesting. So here you have a picture of Laurea tridenta. It's also known as chaparral. It's also known as creosote bush. It goes by a whole lot of different names. You'll find it in a lot of southwestern America, Texas, Mexico, and Caribbean. All right. Very, very powerful herb, and very, very powerful fumes too, almost tarish. Now remember, I said guaiaretic acid was in the lignum vitae tree. Lignum means wood. Vitae means vital or sacred, so it means sacred or vital wood. Okay. Now, this is nordyhydroguaiaretic acid, also known as NDGA. When words get too long, they abbreviate them. They either do acronyms, you know, or they just do initials. All right, so remember I said it's two phenols and their side chains. So you see you got the phenol here, you got another one here, and their side chains came together. So now you have this one dimer. All right, so what role does NDGA play here? Okay, so it is a viral antagonist. Okay, so an agonist is something where you have a receptor and an agonist binds with the receptor, and in it binding with the receptor, it creates, you know, a certain signaling cascade from there. It starts, you know, it ignites functionality, it gets things popping. An antagonist is something that binds with the receptor site and inhibits and shut things down. Okay, it prevents the forward movement, the signaling cascade, you know, from going forward. So NDGA is a viral antagonist. See, if it was an antagonist to you, that means it'd be stopping things that you got going on. But a viral antagonist meaning it's stopping what the virus got popping. Okay, now what does it do? It prevents SP factors from binding to their cognate site. This prevents RNA polymerase from attaching to the promoter. What the hell is he talking about? Okay, so let's talk about that. All right, let's talk about it. I'm going to say it again. A viral antagonist that prevents SP factors from binding to their cognate site. This prevents RNA polymerase from attaching to the promoter. All right, okay, so let's talk about it. When you're dealing with a viral infection, bacterial infection period, you're dealing with a mechanism that their number one goal is to suppress your signaling systems, your immune system, so you can't fight back, so you can't stop anything from happening. You see what I'm saying? It wants to put you to sleep. If you hit with chloroform and somebody gassed your crib, they could come in, clean you out, and you'll wake up like you took a nap and be like, bro, what happened? You see what I'm saying? They got you out the way. They want to rock you to sleep. So there are different protein mechanisms that facilitate the manipulation of communication systems. If you notice in the top picture, all these different cells, we're talking T cells, T helper cells, eosinophils, neutrophils, killer cells. All these cells communicate. The little red circles represent communication, signaling. They're signaling to one another, okay? But what herpes does is it inhibits those red balls from getting to those spots, so those cells don't know what's going on. They don't even know anything popping. They don't even know the herpes is even there, so it can move around and do what it needs to do. When the cat's away, the mice will play, right? So we got ICPs. These are all infected cellular proteins. These are what they call. So you got ICP0, ICP4, ICP22, ICP27, ICP47. You got ICP6 and ICP8. And there's a lot of ICPs, and they all represent something different with the herpes. With ICP0, you got the transcription of many viral and cellular genes. It acts synergistically with HSV1, right? Immediate early protein ICP4, and is essential for the reactivation of latent herpes virus and viral replication. So when I was talking about the nerves before, ICP4, the main boy though, okay? has 8 SP1 binding motifs, promotes transcriptions. The alpha ICP4 promoter in all herpes simplex strains contains 8 SP1 binding motifs, thus providing specific targets for what? This lignin, okay, which I'm going to explain shortly. It's able to bind to the ICP4. If it binds with the ICP4, if it manipulates the ICP4, that means that you're dealing with the alpha stage, okay? You're dealing with the initial. So if you deal with the initial stage, there is no intermediate, there is no late, okay? ICP-47 secretes chemicals that interfere with CD8 T cell modulation. So the top picture, ICP-47 is on top of that. You actually see it in the bottom picture too, ICP-47 herpes simplex virus complex right there. You have a proteasome, which is the orange cylindrical shape, and you see the long black string entering into it. That's a protein that is getting degraded or broken down into peptides, into individual components. Then peptides are supposed to enter through that channel normally so that the signaling to the T cells and the immune system can move forward. But if you notice on the ICP-47, there's a big red X, and what it does is it prevents those peptides from being able to get in through there. So there's no signal. There's no way to know there's a fire without the alarm. There's no way to know, you know, without some type of signal or communication mechanism, and ICP-47 do that. Now ICP-22 and 27, they deal with apoptosis, okay? So 27 is a silver bullet, all right? It plays a role in apoptosis modulation. So you, just like anything else, you're going to have genes for apoptosis. Apoptosis is programmed cell death. This is when the mitochondria is able to make an abundance of superoxide radicals, okay, reactive oxygen species, reactive nitrogen species. All of these species are there to create hydrogen peroxide and pretty much cellular bombs that cause the cell to explode, to blow up. So they actually have infected cellular proteins to inhibit that in them, inhibit that in us. Because if we want to destroy the herpes, we just blow the cell up. If I blow the cell up, I'm gonna blow all the contents up inside. So if the killer cells know that this cell is just, oh yeah, there ain't no turning back, boom, mark that thing for apoptosis. Caspase gets ignited, gets lit, and then guess what? Gonna blow the cell up, and then blowing the cell up, blow the virus up. But if you inhibit apoptosis, then guess what's gonna end up happening? Then you're not going to have a situation where the cell can explode, where the cell can be destroyed, and it can keep going. So all these infected cellular proteins right here, there's more than this. This is just some of them. They manipulate the signaling of your immune system as well as the activation of your antioxidant pathways such as your NFR2 and your DJ1. All right. So Nordehydraguaric acid is a lignin. It says prevents SP factors from binding to their cognate site. This prevents RNA polymerase from attaching to the promoter. So this is what we're about to get into, so I can give you a better explanation of what this means. So a viral antagonist, right, that prevents the SP factors from binding to their cognate site. All right, so if you look at the white box, all right, that says A and B, it has two DNA helixes, all right? On this DNA, you have a complex of transcription factors. Transcription factors are a collection of proteins that kind of huddle together. You know, you look at a football game and things is heavy, so they call a huddle. They have to switch the play up and all that. They got to like inform everybody on the team what this new play is going to be. Okay, so that's what the transcription is. A huddling of proteins and football players coming together, and they're like, yo, we about to transcribe something for some hemoglobin. You know what I'm saying? We need more hemoglobin popping. They like bet. They hit all the different points. If you notice, there's the oval says RNAPOL. That's RNA polymerase. So polymerase is part of this construct. So upstream from this DNA, going to the left side of the screen or off the square, you're going to have a region where you'll have these things called enhancers. And the enhancers actually fold over and touch these particular transcription factors, and when they touch, the RNA polymerase take off. I liken it to, I don't know if you used to look at Looney Tunes back in the day when they would have little animations of the dog races. And in order for the dogs to take off, they had this little mechanical rabbit on a track, and it would just shoot off. And then when a mechanical rabbit would shoot off, the greyhounds would go chasing the rabbit. So it's like as soon as the enhancers hit that transcription factor, your T, babe. You got it? I forgot I put it on, sorry. So the transcription factors end up telling the RNA polymerase where they wanted to start transcribing at, where they wanted to end at. Okay, so this region right here where it starts at, where the transcription factors are at, are called the promoter. Okay, the promoter. All right, now the area where the RNA polymerase stops at is called the terminator. Very easy to remember. One promotes, one terminates. Now in this promoter region, if you notice, it says TATA on the bottom. And this is called a TATA box. A TATA box is just an area that's saturated with thymine and adenine, and it just plays a particular role in regards to where to start at, because those repeated units ain't really going to translate into any type of protein. Is there as a mechanism, a signaling mechanism, the same way when you're about to take a race, you got lines there. You stand behind that line. You have lines separating you from each runner. You see what I'm saying? And those lines and that geometry gives you information letting you know where you start at and what lanes you stay in. So it's similar to that TATA box. Now the GC box is saturated. If TATA is thymine adenine, then the GC box is saturated with what? Guanine and cytosine. Now right there at the guanine cytosine rich box, you see something called SP1. Okay, SP1. All right, SP1 is a particular protein that has zinc fingers. And I'm like, what's a zinc finger? A zinc finger is when you take a protein that has two anti-parallel beta sheets. Remember, when you're making a protein, you have a string of pearls, okay? String of pearls. And then you end up folding those string of pearls up into a three-dimensional structure, okay? So you start with the string of pearls, then you got a secondary structure, you got a tertiary structure, and then you have a quaternary structure. All right? Now, the beta sheet and you got the helix, you got the beta sheet, and then you got the final. So the beta sheets are anti-parallel, meaning one is going in one direction, and then the other beta sheet is going in the opposite direction of whatever that initial direction is, and then they have one helix. All right? Then they have these particular amino acids that make up, right, the helix, obviously the beta sheet, because proteins made of amino acids, and there's a zinc molecule, right? Based on the zinc's charge, it attracts these particular amino acid residues that are inside the protein and causes a conformation of shape. And that shape that it makes is these little things. They kind of look like the Tayit structures of Kemet, that represents Aset. Aset had the Tayites. I talked about it dealing with the magnetic flux lines. I talked about it in Black Panther. But if you look here at these zinc fingers, they look just like them. So each circle would represent a different amino acid, but specifically the cysteine and the histidine residues of the amino acid chains are what the zinc binds to, and it buckles or bubbles the loop. Okay? So the viral gene transcription machinery of many family viruses is made up of complex structures containing a scaffold of multiple proteins and transcription factors interacting with each other and with the viral DNA to promote viral gene expression. Specificity protein 1, SP1, is a crucial transcriptional regulatory element controlling more than 1,000 genes and is found at one or several motifs in the promoters of several family viruses. HIV contains 3 SP1 motifs, which directly and indirectly interact with the HIV TAT, okay, protein to exponentially increase the virus gene transcription and replication. HPV has 1, okay, so HSV, herpes, has 8, HIV has 3, but HPV has 1. All right. The creosote bush lignans, the NDGA, the nordiehydroguairetic acid, the dimer of the 2 phenols yielding the lignin, okay, from the creosote bush, from the Luria, all right, have structural determinants that bind to DNA sequences rich in cytosine guanine boxes, and SP1 is likely to be the primary target for these compounds. Because look right here, this is where the SP1 is, in the guanine cytosine rich box. Okay, so the blockade of this sequence by the plant molecule prevents SP1 factors from binding to their cognate site. All right, as a result, the virus replication is abrogated. Thus, these lignans are able to specifically inhibit the gene transcription and the gene transactivation. All right. So going back, the nordiehydroguairetic acid, the NDGA, is able to affect the promoter region of the transcription factor complex, which does what? By it inhibiting the SP1, it is then in proxy inhibiting RNA polymerase from copying any file at all. So if no file got copied, if no blueprint got passed to the contractor, the ribosomes never get instructions to build. And if they ain't building nothing to be built, ain't no virus. All right. So that's 1. All right. Molecular studies show that all HPV types contain 1 SP1 motif in their HPV 16 long control region promoter. Their promoter region is mad long, it's a real long one, which makes them a specific target for the creosote bush lignans. Okay, indeed, MAL4, which is a form of the lignans where they manipulate the methyl groups. Methyl group is a group of CH3, carbon and 3 hydrogens, and they used methylation, which you use on your DNA to open and close. That's how histones work. Histones work through the acetylation and deacetylation of the DNA. That is how, by adding these little groups on, it either will make the DNA like open up or close up so certain things can be expressed. All right. Now, indeed, MAL4 has been shown to interfere with HPV type 16 early promoter P97 to suppress HPV replication via interaction with SP1 transcription factor in the P97 promoter. So in the P97 promoter, the SP1 transcription factor gets suppressed based upon the implementation of the lignans of the Luria tridenta bush. All right, HIV has 3 SP1 motifs, very rich in CG boxes. All right, here's another one right here. Okay, transport associated with antigen processing, or TAP, represents a mechanism that deals with what? The signaling to T cells. All right, so viral immune evasion. What does that mean? You know what an invasion? Invasion is when all the aliens is coming in, all of them are coming in, taking over. You see what I'm saying? And evasion, and immune evasion is when they what? Evade your immune system by blocking TAP function. The cytosolic protein, here go that ICP47, right, from HSV, blocks peptide binding to TAP. So as I showed before with the proteasome, here's another picture with the proteasome breaking it down into peptides. The peptides normally would go in and have a signaling cascade, When ICP-47 is present, it prevents that from even happening. So guess what? Them T cells don't even know you getting banged on. All right. So we just talked about how to manipulate the whole replication cycle, how to prevent that virus from getting in your cell at all with them surface proteins from way back when we was talking all the way back here. You know, when I was back here, I showed the different stages, you know, of how they get inside. So we want to figure out, how do I stop that from happening? How do I stop the unification of the glycoprotein surface and the surface of the host cell from allowing the perpetual movement of the signal, which will then bring the virus inside the cell? How do I stop that from happening? Well, that's when I get into, let's get into the next guy, okay? And the next guy is to explain to you heparin sulfate. So when you think about synovial fluid, right? Synovial fluid represent the viscous, okay, semi-gelatinous, oil-like substance that is in all your joints, your knuckles, your elbows, your knees, okay? This is the mechanism that prevents bones from rubbing together. When you deal with arthritis, when you deal with inflammation with the bones, it's because your synovial fluid got compromised, the viscosity has changed, it's a lot more liquid now. Because it's more liquid, more watery, and the charge has changed in the system. Now you can have cartilage rub on cartilage, that wears down, that creates heat, that creates inflammation, bone starts rubbing on bone, okay? You don't want that to happen. The mechanisms that keeps the viscosity and the charge distribution correct inside the synovial fluid are these particular bottle brush shaped proteins that exist. Okay, you got hyaluronic acid, which I know you hear about that all the time with collagen and looking young again, right? Which creates this, you know, this long protein, right? And then you have these core proteins that extend from it, and then from the core proteins you got these little bristle brushes, and these bristle brushes are made out of heparin sulfate. Not heparin sulfate, but they're made out of the other sulfate molecules that end up making it up. Now, a sister or cousin to those particular sulfate bristles is this, which is heparin sulfate. All right, and as you see here, it has this link, you know, of these particular carbon cyclic molecules, right, with sulfur attached, and these heparin sulfates kind of create a perpendicular attachment to core proteins extending outside the surface of the cell. And that is what the particular glycoproteins attached to. The glycoproteins on the surface of the viruses attached to heparin sulfate on the surface of the host cell, and through that attachment, the signaling occurs that then allows the fusion of the membranes to happen so that the capsid can now enter inside of the cytoplasm of the cell, and we're trying to stop that. So how do we stop that? We stop that with C3. Okay, we stop that with Chondrus crispus. We stop that with the red algae. Now, there's been information that has come out before on Chondrus crispus specifically not being the best thing for herpes, for HSV, you know, being because the arginine levels in C3 are higher than the lysine levels. And if you're trying to have a successful situation, you know, getting rid of the virus and overcoming the virus, then you want to manage, as I said earlier, your lysine and arginine levels. So if the arginine levels is higher in the C3, then why would you take it for HSV? Well, that's because its other mechanisms that is contained is a lot greater. So the only thing I do is just make sure that you add components, like in my C3. My C3 has other components in there. It's not just Chondrus crispus, it's Chondrus crispus concentrate. And then I have some other mechanisms that raise some things up. You can add components to mechanisms that have high arginine to bring the lysine level up so the arginine level doesn't outdo it, okay? Now, if you go to ktthearchdegree.com, you will find the C3 there. Get your own bottle, you know what I'm saying? This is so you can have a cup of tea, 2 cups of tea every day for a month. But if you really want to get on it, if you really want to feel it, then you want to get like a 4-pack, because you want to take one of these a week. You know what I mean? You want to run through this every 7 days. You know what I'm saying? That's my advice. All right. So What you're looking at right now is C3 Chondrus crispus concentrate. For those of you who don't know, Chondrus crispus is an algae, 3 different algae similar to, you know, red, black, and green flag. You got red, brown, and green. Okay, you got the red algae, the brown algae, the green algae. The green algae would be like chlorella. The brown algae would be like kelp or bladderwrack, while the red algae would be something like Chondrus crispus. And in this bowl, you see some freshly harvested Chondrus crispus with its colors just busting out the seam. And what makes Chondrus crispus so powerful is the fact that when you look at the word itself, it says chondrus, as in cartilage, as in collagen, okay, as in the tissue. And then you also have crispus, which means curly. So you have curly cartilage, because when it dry, that's what it looks like. It looks like curly pieces of cartilage, right? Now, what makes Chondrus crispus so powerful is the fact that inside the membrane of their cells, the composition of the membranes is that of carrageenan. And I know you've heard that name before, dealing with neurotoxins and additives. But carrageenan, there's different types of carrageenan. They can isolate it. They can definitely be toxic. But the natural carrageenan that you find inside Chondrus crispus is not. It's actually the most nutritional part. All right. Now, here we have a paper. You know, I do a lot of presentations. I don't cite a lot of information because I do all the research. I do it all, you know, and I put it out there, and I give people keywords, key terms, so they could look it up and they could see for themselves, you know, the road that I went down. However, from time to time, you know, I'll put some papers up and I'll show something. But here we have enzyme-assisted extraction of bioactive material from Chondrus crispus and Cogium fragile and its effect on herpes simplex virus specifically. Okay, several seaweed polysaccharides. Poly means 3 or more, saccharides means sugar, such as carrageenans, sulfate proteoglycans, dextrin sulfate, alginate polysaccharides, and sulfated fucins have been shown to exhibit antiviral activities against human papillomavirus, influenza A virus, and human herpes virus. Additionally, UVA photosynthesizers from seaweeds have also been identified as virucidal against RNA and DNA enveloped viruses. So when they say UVA photosynthesizers, they're talking about the mycosporin-like amino acids, which are these mechanisms that you find inside Chondrus crispus that kind of give it its darkness, because they grow in the Arctic oceans, okay, in the cold waters, and they're exposed to high level of UV radiation, and in order to deal with that UV radiation out there in the ocean like that, they have their own kind of melanated state where they create these amino-like, you know, these amino-like amino acids that have UV manipulating capabilities in them, and they're trying to say that those mechanisms inside the phycobilioproteins and the mycosporin-like amino acids of the Chondrus crispus both add to their antiviral effects. However, it's really the carrageenans that's doing it. All right, so let me explain. Chondrus crispus, kappa, and iota carrageenan interferes with the glycoprotein heparin sulfate connection of HSV variant fusion. Polyanionic compounds. When I say polyanionic, okay, when you say an anion, you're dealing with negative. When you do a cation, you're dealing with a positive. So when you're dealing with a polyanionic compound, I mean a compound whose growths or endings or whatever are predominantly negatively charged, okay, they're going to interact with the positively charged amino acids in the surface protein of the virus envelope. So the glycoprotein extending from the virus envelope, right, the amino acids that make up their protein that are charged, they're positively charged, okay. Then you got polyanionic compounds that interact with that in order for them to come together. What they're saying is sulfated galactans. Sulfated galactans are the carrageenans of the Chondrus crispus membrane. Okay, sulfated galactans and external glycoproteins of HSV leads to virus inactivation, blockade, and virion binding to cell receptor during adsorption. Adsorption. Absorption, you got a sponge, you got a paper towel, you got a rag. You soaks them up. It absorbs it. Adsorb is when particles attach to a surface. Okay, so maybe I have a cup of water and I have magnetic particles at the bottom of the water, and I take a magnetic stick and I stick it in the water, and those little magnetic particles attach to that's adsorption. Okay, so what's being adsorbed? They're talking about the adsorbing of the viral body to the surface of your particular cell. But in this case, the sulfated galactans, aka carrageenan, aka Conjus crispus components, C3 all the time, all right, interrupt that connection and prevent that from happening. So if it can't get in the cell, replication don't even got to get discussed. You hitting it from the one. All right, so these are different carrageenans. You got kappa carrageenan, iota carrageenan, lambda. I got a better one. Actually, let's bring this guy. Kappa, iota, lambda. All right, kappa, iota, lambda. Now, for those of y'all that wonder why your purple sea moss don't gel, the reason why purple sea moss and certain sea mosses do not gel is because of their concentration of kappa, iota, lambda, and all the other type of carrageenans that exist. Kappa and iota carrageenans specifically are the carrageenans that are responsible for the gelling. All right, and as they gel, they create helixes. The helixes create coils, and then the coils intermingle, and as they do, they build up and they gelatinize, able to store water. Okay, electrochemical gradient and charge distribution of helix and coil induces gelatination. All right, these images are images actually of to give an idea of starch, but these are here so I can actually show what the chain links look like. So when you take these particular molecules right here, these are chair-shaped molecules. All right, normally you got a hexagon shape, but if I fold one end of the hexagon down and one up, then I'll make a chair because they're on different planes. All right, and when you take these particular molecules and link them together, they make a chain, and these chains are what you're seeing right here, just to give you kind of idea of how they're all molecularly, you know, linked up. All right. Okay, so once again, why is Conjus crispus so important? What is the importance of Conjus crispus? It's the fact that the heparin sulfate bristle sticking out the core protein on the surface of our host cells, or what are responsible for binding with the glycoprotein on the surface of the viral envelope, okay, of the viral envelope. All right, this attachment right here. All right, so what the carrageenan does, which looks just like the heparin sulfate, if you look at this particular molecule right here that makes up the heparin sulfate, right, it almost looks exactly like the carrageenan molecules, all right, in their chain. So that's why it's able to inhibit, it binds and it blocks. So guess what? Now the herpes can't get in. Now the virus can't get in your cell. All right, so we're about to wrap it up. This right here are 3 examples of neurons, all right, inside the brain. Okay, you see the blue one, then you see how the color's changing. All right. The reason why the color's changing is because they are deteriorating. Okay, specifically you see these little bodies that's on there, that's amyloid plaque, and when we come back, I'm going to tie how Alzheimer's has something to do with herpes and other viruses. So a healthy neuron, as you see all the way on the end, you notice that these little yellow little bodies that are on them, they show that each corresponding neuron is breaking down and deteriorating. So you get all the way to this brown one, and you have actually got inflamed particles on there tearing it up. All right, so let me give you an understanding of Alzheimer's and show you how herpes plays a role in it, okay? So what you see here is a picture of a neuron, and the yellow kind of like straw represents the amyloid plaque. That is one of the main things responsible for Alzheimer's. And for those who don't know about Alzheimer's, if you haven't been around, it deals with memory. You know, you can't remember stuff, remember people's names, putting things together, you're thinking, everything's getting tore up. And that's because of the inhibition of signaling and communication and circuitry in the brain. If I want to make a call to somebody on the phone and I get to an area where there's no signal, it don't matter how much intention I have to tell this person this information. I could be just so lit that I want to tell this person information. It don't matter how excited I am. It has nothing to do with the fact there's no signal for me to send the message. So the amyloid plaque prevents signaling. So no matter how much intent your nerves, your neurons have in doing its job, it'll fire, it'll send the info, but it's not gonna make it because it gets insulated, it gets prevented, it gets inhibited by the plaque. So we start with something called APP. Yeah, you know me. Amyloid precursor protein. Okay, it resides on the molecular surface of neurons. Proteins form a lattice on the intracellular side, okay, of the cell. This creates a vesicle encapsulating the amyloid precursor protein. The lattice disassembles and a vesicle is transported to the endoplasmic reticulum. Enzyme known as beta secretase cuts amyloid precursor protein extended out from the membrane, leaving a stub in the membrane. Another enzyme, gamma secretase, cuts stub in two. Usually the stub gets broken down. However, in unhealthy cases, they accumulate, forming fibers, and fibers aggregate to form tangles of fibers referred to as plaque. So here I have them in sequential order, and you're going to start from the upper left side of the screen, okay, go down, then go to the middle column to the top, go down, then go to the right column at the top and go down, and these are going to be the different steps. So amyloid precursor protein resides in the molecular surface of neurons. Boom, right? So that's the first picture. All right. Then you have then proteins form lattice on the intracellular side. All right. So if you look at the second picture, we're now on the intracellular side. First we're outside the cell. Now we're inside the cell. You see the little hook-shaped proteins coming there. Okay. Then this creates vesicle encapsulating that protein. All right. So now you see the actual spherical shape that the protein made, and it actually creates a vesicle in the process out of the membrane that's now holding what? The protein inside. All right. So then the lattice disassembles and the vesicle is transported to the ER. So the vesicle the actual proteins that made the little sphere shape break down, the vesicles left behind. And if you notice in the middle, the black, it's traveling down the axon going to the soma so it can go in the endoplasmic reticulum. Under that, now it's on the surface of the endoplasmic reticulum. All right. Now, the beta secretase cuts the APP extended out from the membrane, leaving just a stub in the membrane. Okay, so just imagine that you cut in a plant. Okay, you didn't pull it up from its root. You cut it at the base where it meets the soil. What's inside the soil? There's still a little piece of stem in the soil, and then there's still root. So in this case, this enzyme just cuts the top part of the plant stem off and leaves the root in there. Then another enzyme comes and cuts the root. Okay, then after it cuts the root, those two pieces, they go off and they usually get further broken down and recycled or used for something. But when you have certain signals, certain situations that pop off in the body, unhealthy, those stubs, those roots, those leftover pieces in the membrane that get cut in two, they start to stack on top of each other and form fibers. Okay, as you see in the second to last square on the right column, okay, fourth down. And then on the bottom one, those fibers then come together to make fibrous structures, brushes that are known as plaque. Now, The interesting thing about that is the fact that your body will do that purposely to protect the brain from viral infection. So if there's viral infection in the brain, an overabundance of microbes in the brain, because there is a brain microbiome, and when they get out of control, that's going to signal your neurons to not disintegrate that protein, and thus do what? Thus create those fibers, thus create those plaque. That plaque is there to encapsulate the viruses, to encapsulate the microbes, okay, to keep them from destroying nerves. Because you don't want your pyramid neurons, your pinealocytes, you don't want them serious nerves in your hippocampus and your amygdala destroyed. That's really going to tear your reality up. So your body has so many mechanisms to continue to protect itself, and that's one of the ways it does it. So a lot of people have Alzheimer's or have a large amount of amyloid plaque within inside their brain structure because they actually have a viral infection occurring going up in there based upon what? Like we mentioned earlier about pleomorphism, the upward regulation of microbial bodies. So based upon your eating, based upon your living, your thinking, the type of poisonous toxic light entering into your brain on a constant day, circadian mismatch, this induces the upregulation of microbes that can then what? Infect your brain. But as a defense mechanism, you create amyloid plaque to do what? Encapsulate them, keep them from doing all that damage. But if it's too much plaque, no memory, dementia, you're dealing with a situation. Okay, so this whole presentation was not to break everything down about a virus. I know there's certain parts I left out and there's more to add to it. But this body of work was done to just give the average individual just a little bit more weight so they can get a better understanding of what they're dealing with or their friends dealing with or what they could be dealing with given that they end up in a situation. So, you know, we talked about the carambola and the star fruit with the whole lysine-arginine ratios. We talked about Laurea tridenta, the creosote bush, dealing with the nordehydraguairetic acid, NDGA, lignans, and the other 11 dibenzyl molecules, as well as cyclolignans that are inside that plant. I only talked about the NDGA, but there's more mechanisms in that plant at work. And then, of course, we talked about the Conjus crispus and the C3 mechanism dealing with the carrageenan and the repeated structures of the galactopyranosyl residues. You have the 4-link and the 3-link, you know, and they end up making these particular polymer change that are utilized in order to inhibit the heparan sulfate glycoprotein unison of the viral coat or envelope and your membrane of your host cell. So it inhibits that. So between the Conjus crispus concentrate or Conjus crispus C3 and other algae, you know, even brown algae help with the fucoidin and the mechanisms that they have in their membranes. The creosote bush, these are all mechanisms that can help, but there are a lot more dietary changes that you need to make, lifestyle changes you need to make, spiritual, mental, emotional, of course, physical, that you need to make in order for you to overcome this viral situation. You know what I mean? So all of this is brought to you by Green Life Botanicals. Okay, please go to ktthearchdegree.com, you know, for any of your tonic, compound, or superfood needs, to stay abreast to, you know, upcoming events, things of that nature. KT the Arch Degree also on Instagram and YouTube, you know, for upcoming events and free information. Green Life Travel and Retreats, okay? Everybody knows taking people to the proper environment now. You know, for those of y'all interested in the upcoming retreats, we have one more in this year of 2018, and we got a couple coming in the year 2019. Y'all need to sign up. Come experience true wellness in paradise. We take you to the island of St. Croix, also known as II. We get cooking workshops, herbal workshops. This thing right here you just got, just face to face and a little bit more customized for you. Culture, food, you name it, beach, you know, just a tropical paradise escape, so you can kind of retune your whole clock and get back ready for the world, you know. So anybody who's interested in having a change of speed, I suggest that y'all get with the program. But with that being said, peace, love, and light. K2theartistry.com. See y'all soon. Peace.